We'll move on to our next presentation is Marissa Zago, and Marissa is a master's student at the Department of Biology at University of Waterloo. Her thesis work investigates important outcomes of wetland restoration efforts at Long Point. I'll now hand the screen over to Marissa. Hi, nice to meet
Hi,
you.
nice to meet you too. Thank you
Hi.
for having me. Okay, hello everybody. I am Marissa. I'm a master's student at the University of Waterloo, and today I'll be talking about my research, which is looking at how marsh birds in a Great Lakes coastal wetland have responded to the removal of an invasive grass species, European common reed, which is also known as Phragmites australis. So my research takes place in Long Point, Ontario, which is on the north shore of Lake Erie. And within Long Point are these expanse and diverse coastal wetlands that provide habitat to a wide variety of species, including many that are at risk. And Long Point is of global significance. It's a Ramsar wetland of international importance, and it is a globally important bird area. But it's also of great regional importance in Ontario, as they are some of the most pristine wetlands remaining in the southern portion of the province, and this makes them really important for local populations of breeding marsh birds. And within Long Point are two national wildlife areas, the Big Creek NWA and the Long Point NWA. And the purpose of an NWA is to conserve ecologically significant habitat and the species that use them. And there are several species, marsh bird species that are species at risk and species of conservation concern that reside in these two NWAs. But since the mid-90s, marsh bird populations in the southern Great Lakes region has been experiencing significant declines. Recent research has indicated that the expansion of Phragmites is a factor that is contributing to this decline. And some studies have found that certain birds have been found in lower abundances or not at all in dense Phragmites patches. So marsh birds tend to prefer a habitat known as hemimarsh, which is emergent vegetation like cattail, seen here on the left, interspersed with open water. And the open water pools gives birds feeding grounds, and the nearby vegetation provides places for birds to place their nests or hide from predators. But Phragmites comes in and displaces this native vegetation and homogenizes the habitat with these dense and tall stands that can actually fill in these open water pools and reduce foraging grounds for many species. And the density of Phragmites may also restrict larger bodied species from maneuvering through it. So overall, Phrag can really reduce the habitat for many marsh nesting birds. So Phragmites has invaded the two NWAs within Long Point, and the Canadian Wildlife Service has begun a project to remove 90% of Phrag by 2025. And this is being done by treating Phrag with a glyphosate-based herbicide, and this is being applied by helicopter. And then secondary treatment will occur with an amphibious marsh master, which is seen here on the right, and it comes in and rolls over and flattens the dead Phrag. So in 2019, CWS completed a small-scale pilot project where they treated two small areas in both NWAs, and this was kind of like a test before they continued on with a larger scale treatment. So this is what my research is focusing on. I'm looking at the short-term response of marsh birds to this small-scale removal of Phragmites. We undertook a before-after control impact study to monitor birds both before and after treatment. And to survey the birds, we used these little recording devices that are called autonomous recording units. And you can deploy them in the field and program them to record at any time. And when you retrieve them, you can listen to them and identify which species are present in each recording. So ARUs were deployed in Phragmites-dominated habitat in the spring of 2019 to record birds before treatment. And 4 were deployed in the Big Creek NWA, which is on the left, and 2 more were deployed in the Long Point NWA. Treatment occurred in the fall of 2019, and ARUs were redeployed in the same locations in the spring of 2021. And we listened to each of these ARUs for 45 minutes on one day in June, and we split this amount of time across the early and late portions of the dawn chorus. And this was because our preliminary research had shown that this would maximize species richness estimates and capture both species of conservation concern and species at risk, which were our main target species. So these are some photos that are showing what the wetland looked like before and after treatment. We can see 2 years after treatment, the habitat remaining is very much open water. There may be some remaining dead stems of Phragmites, and there is a bit of vegetation rejuvenation happening. You can see that there's a lot of floating and submerged vegetation with a bit of sparse, robust emergent vegetation. So now I'll talk about some of our preliminary results, looking at if treatment impacted total species richness and the composition of the bird community. For total species richness, we ran a two-factor ANOVA to test if the interaction of urine treatment had a significant effect on richness. And our results are at the margin of significance. You can see that the p-value is relatively small, but it's not at that 0.05 level. But if we look at the box plot, comparing richness in the 2019 and 2021 sites, we can see that it looks like total species richness experienced a greater reduction in treated sites compared to the control sites. And it's possible that species richness declined because of the large expanse of open water that remains after treatment and the lack of vegetation that has grown back over the 2 years. So open water may not be suitable for larger bodied species like ducks and herons, open water may be more suitable for species like ducks and herons, but it may not be as suitable for many smaller bodied species like the red-winged blackbird or the marsh wren that do typically require more dense vegetation for breeding and foraging. So the removal of frag would have displaced a lot of these smaller bodied species, which would have reduced richness tallies in the treatment sites. And it will be important to continue to monitor these bird communities to see if richness continues to decline or if it slowly starts to increase again once the vegetation starts to regrow. So we then looked into community composition to see if control and treatment sites supported different bird communities. So we calculated a non-metric multi-dimensional scaling plot to visualize how composition may have changed over time. And on this plot here, just trying to show that control sites are indicated by squares and treatment sites are indicated by circles, and they're color coordinated to show that the blue sites are frag and the red sites are treated. And this is just a reminder that the initial treatment sites weren't actually treated until the fall of 2019, so they were still Phragmites. And what an NMS plot shows is that sites that have a similar bird community composition will tend to plot closer together, whereas sites with dissimilar bird community compositions will plot further apart. So we can see that the Big Creek sites kind of clump together and are on the left side of the plot, whereas the Long Point sites clump together on the right side of the plot. And from this, we concluded that the two NWAs do support distinct bird communities. And just a reminder to look at the map, we can see that Big Creek is on the west side of this peninsula. And the ARUs that were placed in the Long Point NWA were placed at kind of the very tip of the peninsula. So these regional differences in bird communities are likely masking our ability to see an effective treatment. So because of this, we decided to look at the Big Creek sites only because they had a larger sample size. And we ran a two-factor ProANOVA looking at the interaction of urine treatment. And when we did that, we found that there was a significant effect of treatment over time on the community composition in Big Creek. So now the big questions, how did community composition change and which species may have been positively or negatively impacted? So some patterns that we're seeing is that it's a bird's life history traits. So how they forage, what they forage for, and where they nest are factors that influence whether they're impacted by frag removal or not. So I'm going to highlight some trends we saw with species of conservation concern and species at risk. And species of conservation concern, they tend to be secretive marsh bird species that typically require a good quantity or quality of habitat, and they tend to be the larger bodied species that may disfavor Phragmites habitat. So over the 2 study years, we observed the 5 species on the left, and there is a hint of a trend that 3 of these species may be positively impacted by frag removal. The American bittern, common gallinule, and pied-billed grebe were present in 1 to 3 of the 3 treatment sites. And these birds tend to forage in open water by stalking, dabbling, or surface diving. And it's likely that the open water treatment or the open water habitat that remains after a treatment would increase their preferred foraging grounds. On the other hand, the least bittern is also a bird that forages by stalking, but it may have been negatively impacted by frag removal. And typically the least bittern requires tall, dense emergent vegetation, which can be cattail or sometimes frag. And they need this to cling onto while foraging. So treatment would have eliminated all the vegetation and may have driven this bird to other areas of the marsh that had a greater amount of vegetation present. But this isn't to say that the least bittern occurrence will always be impacted by treatment. The vegetation, again, will need time to rejuvenate, and the habitat requirements of this bird may be met again in several years. So the barn swallow and bank swallow, which are both threatened species at risk, they also show a hint that they may have benefited from treatment. They likely gained foraging grounds because they are aerial insectivores that forage over open water. So some species were negatively impacted by frag removal. These tended to be the small-bodied species that are foliage gleaners and shrub nesters that do actually use Phragmites for habitat. And they use this because frag can supply them with an adequate amount of insects, and its vertical structure is really beneficial for them to move around on and place their nests in. So when frag is removed, these birds tend to lose some of their foraging and breeding grounds. But it is important to note that a majority of these species that were negatively impacted by frag removal, they tend to be habitat generalists, meaning that they don't solely rely on marsh as their main habitat. So it's likely when frag is removed, they can go off to other habitat types like forests to nest and forage. So what we've seen 2 years after treatment is that there may be a change in species richness, and there is a change in community composition. It looks like the larger bodied species that Phragmites may have been previously excluding are now benefiting from the open water habitat, while the smaller bodied habitat generalists that do use frag are now displaced. But these trends aren't permanent, and they will continue to change. Future monitoring is needed to see how the vegetation will regrow and how birds will continue to track these changes in their habitat. So thank you all for coming to this presentation today. I'd just like to acknowledge my peers who helped with the data collection for this project, as well as my supervisor, Dr. Rebecca Rooney, and everybody else in the Waterloo Wetland Lab.